Okay, guys, so very quickly, I wanted to talk to you about protein synthesis. And this is an overview of protein synthesis. So, protein synthesis is basically that formation of the polypeptide, that primary structure of the protein. And this primarily happens in a ribosome, and it happens in two stages. The first stage is transcription, and this occurs inside the nucleus. So, transcription is the process of creating messenger RNA from DNA. So, this is where one of the genes of in the entire nucleus is being transcribed into messenger RNA. The reason we create a copy of the gene of interest into RNA is twofold. So first, the DNA chromosomes are just too big to leave the nucleus. They can't get out through the nuclear pores. And second, we don't want the DNA to leave the nucleus because in the cytoplasm of the cell, there are lots of enzymes that would destroy the DNA. So we want to keep the DNA protected inside the nucleus. So instead, we make a copy of the gene of interest, and that copy is in the form of messenger RNA, which is allowed to leave the nucleus. So because we're only making one copy of a gene in the genome, and humans have approximately 23,000 genes in their genome, it's small enough to exit through the nuclear pores and go into the cytoplasm and attach to the ribosome where it will be later transcribed. So in order to begin this process, RNA polymerase comes in to the double-stranded DNA and it unwinds the double-stranded DNA and it moves along this. So here's my RNA polymerase enzyme. It's unwinding the double-stranded DNA and it moves along this DNA in a 5' to 3' manner. So that means that this messenger RNA, if I look at the tail is always going to be 5', and the head, where I'm adding my RNA nucleotides, is always going to be 3', because the ribosome is moving in the direction of 5' to 3'. So it is hydrogen bonding these nucleotides in place and covalently bonding the sugar phosphate backbone to the growing messenger RNA strand. The strand that it's making the copy from is called the template strand. Another name for the template strand is the antisense strand. So it's positioning these nucleotides on the template strand with complementary base pairing, so we get an exact copy. Then it's linking the RNA nucleotides by covalently bonding that sugar phosphate backbone. And then when it's all done, it detaches from this assembly, and the RNA is set free to where it will undergo other modifications. So just a quick mention of the two main differences that we see between DNA and RNA. Recall that DNA is double-stranded and it's anti-parallel. So you have your 5' to 3' end, you have your 3' to 5' strand. Also recall that RNA has uracil rather than thymine. So recall, just like DNA polymerase, that RNA polymerase is adding the new nucleotides, those RNA nucleotides, to the 3' end of the growing chain. As a result, transcription is occurring 5' to 3'. So if I take a closer look at this, here you can see my chain that's growing. If I move my new free nucleotide over here, I'm going to be adding this 5' phosphate to this 3' carbon right here. So I'm going to keep growing 5' to 3'. So in prokaryotic cells, RNA is translated as soon as it's transcribed, because remember, prokaryotes don't have a nucleus. So as soon as the RNA is transcribed, it's being translated. Whereas in eukaryotic cells, we've separated it out into translation, which is what happens into transcription, which is what happens in the nucleus, and translation, which is what happens in the cytoplasm. Because these two functions have been separated out between the nucleus and the cytoplasm, it allows for a significant amount of post-transcriptional modification. In other words, after this is transcribed, we're going to process it, and that's what you see here. So what do I mean by post-transcriptional processing? And what we mean by post-transcriptional processing is we add a 3' tail, a 5' cap. We take out things called intervening sequences, and we leave in express sequences or exons. So what do I mean by my 5' cap? Well, what you can see here is I have my messenger RNA being built right here. So this is my messenger RNA in purple. And then if I look over here on this side, what I see is I see a GTP, which is my guanine triphosphate, which you can see right here. Here's my guanine, my sugar, and my triphosphate. So it's an extra GTP, but it also has this methyl group right there on the GTP. And what that does is it literally is acting like a hat or a cap put on the 5' end of my messenger RNA. And if I go down to my 3' end of my messenger RNA, what I see is I see a polyadenylated tail. That means I put a whole bunch of adenines at the end of it. Both the cap and the tail help to stabilize my messenger RNA in the cytoplasm and protect it from the degradation by enzymes. Eukaryotic genes that code for messenger RNA have two types of sequences in their genome. They have introns and they have exons. So here in pink, you see the introns, and the green are the exons. And so these introns we refer to as intervening sequences, and they are removed. They can serve multiple functions, which we'll talk about in a little bit, and the exons are then put together, and that creates my messenger RNA, which is going to have the cap and the tail. And then we'll leave through the nuclear pore into the cytoplasm. So one big advantage of having these alternative splicing means that I can put these exons together in different orders. And by being able to put them in together by different orders or leaving some out altogether, what I can end up doing is I can make different proteins. So here's an mRNA that's translated into protein A. And in this sequence, you see you have exon 1, exon 2, exon 4, and exon 5, and it's translated into another protein. And in this one, protein C, what you have is exon 1, exon 2, exon 3, exon 5, and 4 is left out, and it's translated into a separate protein. So all of these genes can be alternatively spliced to make different proteins. So I save in my length of genetic code required to make a protein. So I'm condensing the space needed for my genetic material. So after we've had our cap, our tail, our splicing, I'm now ready to leave. But remember, this only happens in eukaryotic organisms. Prokaryotic organisms have no post-transcriptional modification. So this is only in eukaryotes. And now we're ready to go to the cytoplasm, where we'll be met by a ribosome and we'll start the process of translation.